You've seen this before. We started with it. What's the central dogma of biology? Central belief system. It is a core foundational thing that we all believe is true.
DNA codes for RNA.
You betcha. And when we said we started cellular, molecular, developmental biology, when we started that, we said that this is at the very heart of the way that all organisms work. So, in biology, you do not have a safe with a lock in your cell. But you do have a very, very safe place. Where is that? That's your nucleus. And all of the DNA in a cell is going to reside there. And it's never going to leave because if I'm borrowing one from AJ, you're done.
Yes.
And in the case of a cell, that's actually true. If something goes wrong in the DNA, it will be dismantled for parts. Don't worry if you lose your birth certificate or your social security number. In the perfect world, you will not get dismantled for parts. Notice I didn't say that you wouldn't. I said in the perfect world you wouldn't. I can't make any guarantees. Oh, it's sad, but it's true. Now all of you are going to leave here being afraid of being dismantled for parts. That DNA is going to be turned into a temporary cheap copy that we call RNA, and it is free to leave the nucleus and enter the cytoplasm. That temporary cheap copy, back in the day, it used to be, what was it with that ridiculous purple nonsense? Ditto. The Ditto machine made a temporary cheap copy, right? You guys have no idea what that is.
Never seen a Ditto machine?
They've never seen a Ditto machine. Truthfully, honestly, I've never seen a Ditto machine.
I'm old enough, right here, because I have used a Ditto machine.
Ooh.
So it is a way of making a cheap copy. It's like before there were Xerox machines.
Mm-hmm.
Cheap copy of anything?
Well, printed back.
You just like put like a desk in there.
You just set it up there.
Yeah. Stop.
So the Ditto machine is in fact how dittos were made, which is why sometimes people still call photocopies dittos. They're not actually dittos. They are photocopies. Today we would use a photocopier. You guys at home would use a scanner, but your cell doesn't have any of those technologies. Instead, it's going to use a process called transcription. And this is actually way more old school than even the Ditto machine. Even transcription outdates Mr. DF. Because transcription is how we used to get copies of things way back in the day, pre-printing press era. Pre-printing press era, typically it was monks who had to do this because most of what we really needed copies of were religious articles and books. They really needed prayer books. So the poor monks would be forced as part of their, monastic duty after they did Vespers, Liturgy of the Hours, did all the stuff that monks have to do. They would have the original copy of the book. They would have it out in front of them. They would take their dip pens and they would dip the pen and write word for word the copies of everything. And it's called transcribing. Scribe means to write. So they were transcribing it from one page onto another page. And you can still go see some of these truly amazing copies in all kinds of amazing places all over the world. And then you can imagine that poor monk offering that suffering up as a part of his vocation. How many of you are like, yeah, sign me up to do that for a living? Robert's like, heck yeah, I'd make no mistakes. I'd be perfect. So that process, this transcription process, is exactly that idea, a copy letter for letter. Once the RNA reaches the cytoplasm, though, it actually has to be used. We have to figure out what it does. That process is slightly different. If we're going with the monk analogy, while it's good to copy holy books of prayer, it doesn't do you much good if you don't open the holy book of prayer and, oh, I don't know, use it to pray. So the process of taking it from the language of words to one of prayer has a different name. We're changing languages from the language of nucleic acid to the language of amino acid. Now, I know you guys are masters of this, especially those of you, my friends, who I think Latin students do this a great deal. You have to take a passage in Latin and make that passage English. What do you call that homework assignment?
Translate.
Yeah, you are given a translation assignment. So the biological process of translation is taking our RNA and turning it into amino acid. And of course, you told me in your Bell question that amino acids are simply the monomer of a protein, right? Of course you did. And you told me how many potential amino acids were there? 20. So we went from a 4-letter language of nucleotide, right? That's how many there are, to a 20-letter language. That got complicated. Got complicated fast. Yeah. With me so far? This whole central dogma thing, we're okay with it? The process of transcription and translation, when we add them together, is protein synthesis. Now, the best part of this whole thing is Mr. DF is just in here like hanging out, visiting for the day because he's been ousted from the library for AP exams. And, well, we love him, so we'll take him. And it's great for those of you who are in data and design because this is the beginning of what we do together. And he gets to be here for your learning of it. Aw, team sport. Yay. I don't know, you already picked a theme song for us. No, Mr. DF. Stick around after, he'll be thrilled to play you this theme song. He will too. So, this whole thing starts at the very beginning with a pretty simple difference. We have to figure out the differences between DNA and RNA, but I'm totally convinced you can do this without me. You don't need me for this. How many of you said double helix for DNA? Fabulous. 2 for the number of strands for DNA. Delightful. A, T, C, and G for the nitrogen bases. Yay. How about deoxyribose for the sugar? Oh, fabulous. We learned something 2 chapters ago. And nucleus for the location. Oh, yay. See, I told you didn't need me. RNA, is it a double helix? It's a single strand. And if it's a single strand, I'm going to go out on a limb and say that it's only 1 strand. Oh, good. Adenine, cytosine, guanine, but it's not it's not thymine. It's uracil. The sugar is ribose instead of deoxyribose. Now, the thing about this whole ribose-deoxyribose thing, you guys know I love taking words apart. So the prefix DE, what does it usually indicate? Now, DI would be 2. How about if a situation devolved into complete nonsense? Went back? How about if the stupid birds in my yard denude the cherry tree as soon as the trees have cherries on them? What did they do? They removed them. They stripped it of them. And how about, I had another one. There's another DE. Come on, librarian, don't you have another DE word?
Nature.
Delusional. No, that's
Destroy.
just erase that.
Deconstruct.
Oh,
Deconstruct.
delusional.
Delineate.
Delete. Delete.
Dilutes and DI, right?
Wait, but like, if you can like undo
Yeah,
delete,
yeah,
like,
yeah.
you just elite? I am.
So wait, you're trying to tell me that D means what?
Take away.
To take away. So DE means to take away or less of. Oxy means oxygen. The only difference between our sugars, deoxyribose and ribose. Ribose has one teeny tiny little oxygen attached to a carbon that deoxyribose does not have. Is one oxygen that big of a deal? In a 5-carbon molecule, one oxygen's a big deal? One atom makes a difference? Let's think of other things that involve the change of one atom. Everyone's favorite H2O, water. We like water, right? H2O2 simply adds one extra oxygen and creates the compound
Hydrogen peroxide.
hydrogen peroxide. How many of you are like, yeah, I'm going to drink me some of that?
I have a joke about that. There's a guy that walks into a bar and says, I'll have the H2O, and the guy next to him says, I'll have H2O too. And the second guy says,
Did I tell you that one? Because I use that one a lot.
I know that one.
Who doesn't love a good science joke? Mr. DF's looking at us like, I don't know why I chose this room.
Gentleman who served life, he would have said, oh, like H2O is a well. Yeah, I know. Not just honestly, you're just confusing the guy.
So, you are absolutely correct in telling me the change of one oxygen is an enormous difference. We already answered this one, so I'm just going to fill that in. Cytoplasm, right? Another between DNA and RNA is there's only one form of DNA, but there are 3 forms of RNA. The 3 forms of RNA are as follows. Why am I here if you already know this? Why didn't you send me home early if you already know this? Like, I could be like sitting on my deck, hanging out, talking to my neighbor's horses.
Do you leave space in between?
You're horsing around.
Notice that the first letter is always a lowercase and the RNA is always an uppercase. Weirdly enough, even when those 3 abbreviations begin a sentence, we do not capitalize the first letter of the abbreviation. It's very disconcerting. It's upsetting to any of you who like to follow the rules and conventions of grammar, but unfortunately it is true. We're not going to learn what they do yet. We're going to learn them as they come because I like to know things. For me, it's better in context, better than a list that we've memorized. But they all are important players in protein synthesis. This is where I'm going to fall back on one of my hobbies. And hopefully you guys will understand why I choose this as an example. I like to cook and bake. And in my cooking and baking, I have accumulated a fabulous number of cookbooks. Some of them are just like cheesy little cookbooks you pick up along the way, and some of them are really good books. And one of them that is a collection of very good books is called Mastering the Art of French Cooking. Mastering the Art of French Cooking is very, very, very expensive. It was like, there's 4 volumes, and I think it was like $80 per volume. So I don't take them in my kitchen. Now you're like, how does one go about using a cookbook if you don't use it in the kitchen? It's pretty simple. I look up the recipe I want, I open the page, and then I copy the recipe and take the copy down to the kitchen. True story, that's what I do every time. But you understand that I only use one recipe at a time, right? Like, if that makes sense. You're like, duh, people don't use the whole cookbook at a time. It'd be dreadful. And you've seen previews for that movie, Julie and Julia, right? Where the girl named Julie cooks every recipe from Julia Child's Mastering the Art of French Cooking, one a day. She only did one a day for a reason, because they're not easy ones to follow. So you do one at a time. What would happen if I only copied one page of a recipe that ran over onto 2 pages? It's not going to turn out. So if I miss the beginning half of the recipe, I miss some pretty important starting instructions, right? And if I decide not to copy the end, I'm never going to know how to finish it. So what information do I use to indicate where the recipe begins and ends? How do you know in a cookbook where it begins and ends?
It was like a header whenever a new.
All right, so there's a header, and it tells you, like, you should probably start here. And it doesn't actually say you should start here, but it's usually in bold and in a larger font. And then underneath it, in italics, it usually says something like, make 6 servings. Agreed? Sounds familiar. How do you know you've reached the end?
The next recipe starts.
When the next recipe starts, and you usually, like, the instructions stop. So there's no more instruction after that, so you can continue. And in Mastering the Art of the French Cooking, it's got like this weird mark on the end of the recipe, like it's like a small graphic that indicates like this is the end of the recipe. You can stop here. Your genes, your DNA, it's nothing but a series of recipes for proteins. You don't use all the recipes at one time, and you need to copy the whole recipe. DNA is smart enough to have a header that says start here for this gene, and it has an ending that says stop here at the end. So we're going to take a look at a picture of that exact thing. How do I know I'm looking at a stretch of DNA? All right, it's double helix. There is a region called the gene region. Sometimes in biology we also call that the promoter region of each gene is usually comprised of a series of thymines and adenines. What letter do we use to indicate thymine? And what letter do we use for adenine? And it's a repeat of thymines and adenines, all in a row, so we call it the Tata box, thymine and adenine box. It's just a repeat that indicates to all of the enzymes involved in this process, you should start here. If you start here, you'll get all of the gene. To start this process, then, we begin at the beginning. Not like bad science fiction books that just drop you in the middle of the story and expect that you're going to figure out where you're going. We start at initiation, so transcription begins at initiation. You know what's going to be great about translation? It's also going to begin at a step called initiation. So we only technically have to learn one set of things. During initiation, an enzyme recognizes the promoter and opens the gene there. Actually, it's pre-gene, so it opens the DNA there. And you're going to be very, very, I'm sure, absolutely shocked to find out that particular enzyme has a name. RNA polymerase. How many of you are like, hey, that word's familiar? No one? Oh, a few of us? What did our friend DNA polymerase do? You're like, but I know the words. Remember, we're scientists. We take the word apart. Take the word polymerase apart, and the beginning of it is?
Complementary nucleotides to the primer.
It's going to attach complementary nucleotides because it's a polymerase. ACE means it's an enzyme. Polymer, it's an enzyme that makes the polymer. So here, the polymer that's going to be made is RNA polymerase. So once DNA has been opened, RNA polymerase is going to attach complementary nucleotides to a short primer that had been attached. Remember the flaw that DNA polymerase had? What was it? We tried to call it not a flaw. We were like, don't judge it. It's not its fault. It's not a flaw. It's just it can only do this.
Read in one direction.
It can only read in one direction. Do you remember what numeric direction that was?
3' to 5'.
It can only read the template from 3' to 5'. You know what's true of our friend RNA polymerase? It only reads from 3 to 5'. You don't have to worry about it, though, if you're like, if it only reads 3' to 5', does that seriously mean that I've got to deal with this leading strand, lagging strand thing? No. And here's why. Only one strand of the open DNA is going to be used as a template. So only one side gets read, and conveniently enough, the side that's used as a template is the one that opens from 3 to 5'. So it's a non-issue. We don't have to worry about that lagging nonsense with its Okazaki fragments, all that tomfoolery. Meh. It's done. It's not a problem in this process. As soon as RNA polymerase begins attaching complementary nucleotides, and our strand is more than just a little baby strand, the next step has begun. Elongation. That's just where RNA polymerase continues down the open strand and continues attaching complementary bases. So if my template strand read 3' ACTC ACGT 5', what will my RNA read? Will it read exactly the same thing?
Oh, the U.
Mm-hmm. It's going to read the U instead of the T. So if it's 3', what's it going to be on the RNA? 5'. If it's A, then we read. If it were DNA, it'd read T, but here it's going to read U. And if it's C, it'll match with. If it's T, it matches with A. That doesn't change. C still matches with A, U, C, G, and T. And if it's 5', then it must be 3'. That is our truly tiny Mind you, most of these are typically thousands of nucleotides long, but per our previous discussion, I am hungry and would like more than grapes. So we're not going to go for 1,000 nucleotides because we'd still be here for a while. Do you agree with that decision?
Yeah.
You're not judging it?
No.
Okay, that's good. Much like I know that I've reached the end of a recipe when I encounter that little glyph, that little picture, RNA polymerase knows it reaches the end of the gene it's trying to copy when it reaches a sequence of the gene called the termination sequence. Sometimes also just called the terminator. No one made any noises. Usually someone makes a noise. There you go. Thank goodness. So it reaches the termination sequence, and that's when it gets to end. Now I'd like to pretend that that's the end of transcription, but it's not quite. There's some editing that has to happen. What falls off of that complex of RNA polymerase and DNA is called pre-mRNA. Of course, you guys know that means before. Pre is always before. That's an easy one. Why don't I ask the easy ones? So it means before RNA, or mRNA. I'm going to take it. You may draw your gene sequence in any colors that you choose. You know, most of you are like, I have a pen in my hand, so it will be monocolor. If monotone works for you, that's fine. Some of you are striping them in opposite stripes. I have no doubt some of you are going to put stars in one and hearts in another. So we have our transcript that we'd copied from our open DNA. We have our temporary mRNA, our pre-mRNA. And the first thing that's going to happen in the editing process, just like what you would do if you were about to go outdoors and it were winter, you would put a hat on. But here, instead of a hat, we're just going to call it a cap. And that does happen. We put a cap on of inverted guanines. And those inverted guanines then don't get incorporated into the final gene product because the ribosomal RNA encounters it and it's like, well, this is just the protective cap. We don't translate this and it skips it. Then, just like you put the cap on your head when you go outside in the winter, you put boots on your feet. So here, it puts a tail on, and we call it the poly A tail. I know you'll be shocked to find out that it is simply made of a sequence of adenines over and 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 over again. It's just a whole bunch of adenine. The last thing that happens is called splicing. When we splice something, that means to cut. We've cut it. We are going to cut the mRNA and cut out parts that aren't necessary and re-tape together parts that are necessary. The parts that are not necessary are called introns. And here I've indicated my introns in blue. So an enzyme just comes right through, snips out those blue things, and smushes back together the green bits that are called exons. And Mr. DF, when we went to that super fancy conference last week, were introns and exons words we heard often?